Hello, everyone. Welcome to this week's M2D2 talk. This week we'll have Chenyu tell us about, and I don't have the title in front of me, but anyways, tell us about molecules and all sorts of things. Chenyu is a second-year PhD student at MIT, advised by Tomi Ekola and Caroline Euler. She's also affiliated with the Eric and Wendy Schmidt Center, the Broad Institute, and her research interests are machine learning, representation learning, generative models, and generally AI for science. And very happy to have her with us this week. If you'd mind sharing your screen and getting started.
Let me share my screen. Okay, thanks for the introduction and the invitation. And today I'm going to talk about my recent work about removing biases from molecular representations via information maximization. This has been accepted by InfoCORE 2024, and this is a joint work with Sharut and my advisors Caroline and Tommy. And feel free to ask questions during or after the talk. So for this paper, we want to solve the problem of molecular representations. And when we see a molecule, they have different, there are many different ways to represent the molecule. People sometimes use 2D graph or use the small strings. And traditionally, people will use some like fingerprint to represent the molecule or to featureize the molecule. And later, with machine learning and deep learning, people start learning some unimodal molecular representations. For example, as in these papers, they learn a contrastive learning based on different augmentations of the molecule graph. And however, this purely 2D structure-based unimodal molecular representations is very limited by the fact that we can always see the case that two molecules have very similar structures, but they can have very different effect in the cellular context. And if the pre-training is only based on the molecular structure, it is actually very hard to figure out which is the biologically important substructure of the molecule and which is the irrelevant part, because we don't really have that information in this data. So because of this, we turn to utilize the high content drug screening data and learn a multimodal molecular representation. As a brief introduction, the high content drug screens is a kind of biotechnology that can screen the phenotypes, different phenotypes of the cells after the drug's perturbation. So briefly speaking, we have for example, we have a bunch of cells, and then the drug is added into the cell. And after a certain amount of time, typically 24 hours, we can screen for different phenotypes of the cells, which reflect the biological effect of the drug on the cell. And these phenotypes include the post-perturbation gene expression or the image of the cell after the perturbation. So this experiments is a good way to help us figure out what is the drug's real effect in the cellular context. However, due to the experimental constraint, we are not able to screen every molecule in the wet lab that we are interested in. And so therefore, we want to learn a multimodal molecular representation based on the drug screening and the drug structure modalities. And if it can generalize to new drug structure, we no longer need to do the experiment in wet lab, but at the same time, we still get access to the information of the potential biological effect of the drug structure if the generalization works well. And there are some recent works directly apply the CLIP model in the text and image domain to the molecular domain to map the molecular structure and the post-perturbation cell imaging data. However, with this kind of data, the generalization to new molecules is really challenging because of the noise and batch effect in the experimental data. And as we know, the batch effect is a very significant challenge in all kinds of biological experimental data, because these experiments are high throughput. We have like thousands or tens of thousands of drugs to screen for this experiment. So it is impossible to do them all in the same batch. And so normally the batch effect is because we need to do the experiment in batches. And for each batch, they are screened by the same person at the same temperature, humidity, etc. And different batches may have their difference because of these technical issues, which is not real biological factors. And this kind of similarities may lead to inaccurate understanding of the real biological effect if they are correlated with the outcome of interest in these experiments. And apart from this batch effect, what is even worse is that when we are doing the experiment, we figure out that it is actually hard to fully randomly shuffle all the drugs across different batches, which means that there are some structural similarity of the drugs within the same batch. And this can be shown by a very simple experiment that we find that the batch identifier is actually surprisingly predictable from both the phenotypic screens, which is the normal batch effect, and at the same time, the molecular structure at a quite high accuracy. And the effect that it is predictable from both the phenotypes and the structure means that the batch is actually a confounder. So for example, in one of the cell imaging data set that we are using, we trained a very simple random forest classifier and evaluate the batch classification accuracy on the held out compound. And for the self-provider features, which is featurizer, some handcraft features of the image, we can see that the accuracy to predict the batch ID is over 0.9. Versus the less than 0.1 is with a random predictor. So it means that the batch effect is significant in the data. However, if we only use the molecular structure and a simple multi-vec featurizer to predict the batch ID, the accuracy is surprisingly about 0.5, which means that there's very strong confounding issues, and drugs with similar structures or similar scaffold are placed on the same plate or the same batch. And we can model this with this graphical model. So there are 3 observed variables and 2 latent representations. And for the 3 observed variables, this XD represents the drug structure. This XG is the phenotypic change induced by drug perturbation, including gene expression or cell imaging. And this XB is the batch number, the experimental batch number. And as I described previously, this edge from XB to XG reflects the normal batch effect. However, this edge from XB to XD reflects the non-random batch assignment and makes this batch number XB a confounder between the 2 data modalities. And further, there are also 2 latent representations that we are learning based on each single modality respectively. So with this graphical model, if we don't consider this batch confounding issue and only learn a model to match these 2 modalities, we can imagine that this batch actually provide a shortcut between the 2 shortcut paths between these 2 data modalities, and the latent representations will include the batch related information as the shared features between the 2 modalities, which is not really desirable because they are not biological. So to solve this problem, we propose to use to optimize a conditional mutual information objective instead of the mutual information unconditioned on batch to remove the biases from the irrelevant attribute of batch numbers. So briefly speaking, we want to maximize the mutual information between the two latent representations conditioned on the batch number. And the intuition of this objective is that the features tied to this batch number XB, they doesn't improve the conditional mutual information objective. And if we further consider the constraint on the representation capacity, the batch-related features will therefore be excluded in the latent representations. However, as we know, to optimize the conditional mutual information is not easy in practice, and people are trying different ways to estimate the conditional mutual information and also to optimize some lower bound of the mutual information, conditional mutual informations. So here we think about the problem in the contrastive learning framework and introducing a little bit more background of contrastive learning and multimodal contrastive learning. So this is a figure from the CLIP model, which is trying to match between the image and their corresponding text caption. And here we see that there are there are individual encoders for each of these two modalities, a text encoder and an image encoder, and they map the data to the latent space with I1 to IN and T1 to TN. And in the CLIP model, they calculate the cosine similarity between each of the latent. And so for example, if we take the I1, this image latent as the anchor, then its corresponding text latent T1 is called the positive, and all other irrelevant text latent is called the negatives. And the CLIP model want to maximize the positive similarity and minimize the negative similarities, which means that in the latent space, we want the anchor and positive to be as close to each other as possible. And we want the anchors to be further away from the negatives. And symmetrically, we can also take the text encoder, the text latent as the anchor, and all other variable and the image latent as a positive and negatives. And more specifically, they are optimizing the influence E loss, where this F is the cosine similarity divided by a constant, like a temperature hyperparameter between the two latent representations. So in this loss, we can see that it is maximizing the positive similarity and minimizing the negative similarities in the denominator. So in these previous papers, they built a nice connection between the influence E loss with the mutual information between the two data modalities. And the intuition is that we can see that the mutual information has a formula like this, and they use the exponential family. They use an exponential family to approximate as the variational distribution of this conditional probability. And for the marginal probability in the denominator, we can see that the corresponding variational distribution is the sum of all the negative similarities. So it is actually to use the average as an approximation of the marginalization. So in this way, we can show that the influence E loss is a lower bound of the mutual information. And furthermore, our problem I described previously can also be viewed in a general framework as a sensitive attribute problem, where here the batch number is the sensitive attribute in our problem. So with this kind of problem, there are some methods developed in other domains, for example, to improve the representation fairness. And there are some previous papers that are trying to deal with this. The first one is a conditional contrastive learning method. Oh, I see a raised hand.
Yeah, sorry, I have just a small question. Yeah, on this graphic, how do you account for unobserved variables? Because I'm sure there's some unobserved latent that a causal feature that impact both ZD and ZJ. Yeah.
Yeah, so here, there are two kind of unobserved features. One is that, so one is those that only affect XD or only affect XG. And because we are learning the shared representation between both, so hopefully from the model, those features will not be learned, in the representation because they are not shared between these two modalities. And for the other, unobserved features, for those that are affecting both XD and XG, we think that apart from the batch confounder, the other features are the biological meaningful features. Because if we think what is the shared part between the drug structure and the phenotypic screening, so what links them is the drug's mechanisms to the drug, and that is exactly what we want to capture in the latent representations.
Perfect, thank you.
No problem. Yeah. So first baseline is the conditional contrastive learning method, where if we think about the conditional mutual information, a very simple way to think about it is to sample the negatives instead of sample the negatives from a marginal distribution. We can sample it from a conditional marginal distribution conditioned on the batch number XB. And then we apply the same influence AE loss as before. And it can remove the batch confounder because if all the positive anchor and negatives have the same batch number value, then it means that the only thing that makes them different is the non-batch related features, which are real biological features. And if we want, so with this, we want to distinguish the positive and negatives with respect to the anchor in the latent space. The latent have to capture the real biological features, and they can't utilize the shortcut of this batch confounder, this XB. Theoretically speaking, they can remove the batch confounder. However, this doesn't fit well with our problem because in practice, in the data sets that we are using, there are very limited samples within each batch, and there are hundreds of or thousands of different batches, and only 200 or 300 drugs within the same batch. So if we only do the conditional sampling, we have a very limited supply of negatives, and we can only utilize the negatives from the same batch, which is not diverse enough, and this will limit the capability of the model's generalization to new drug structures. And there are also some previous work that use some generative models to generate an unbiased image training data in the computer vision domain for fairness. However, in this problem, we have to generate the counterfactual samples of the drug on another batch for the gene expression or the image. And the generative models in this domain is not very well developed. And also it's very hard to evaluate the generated image or gene expression. It's hard to validate that they are reasonable generative samples. So this previous method don't fit well with our problem. And to solve this problem, we propose reweighted influence objective as an alternative tractable lower bound for the conditional mutual information. And here there are several terms in this lower bound. The most important ones are the first two terms. And the first term is shown here. We can see that it is very similar to the clip loss as shown in the top right corner. And the only difference is that it has this two posterior batch distributions as the reweighting factor framed in blue color. And this posterior batch distributions is the batch distribution condition on both the anchors latent and the negatives, positive and negatives latent. So intuitively speaking, this reweighting factors is adjusting this influence AE loss, and it is giving the negative samples with more similar posterior batch distribution to the anchor a higher weight, and give the samples with a lower, with the less similar posterior batch distribution to the anchor a lower weight. And with this, we can imagine that for the samples that have a more similar posterior batch distribution with the anchor, it means that the latent contains similar batch-related features in the latent representations. So it means that when they are when we use them as a negative sample to contrast the learning of the anchor, it will have better contrastive capability because they share the same batch-related features, and the model can't use batch-related features as a shortcut. So it needs to learn real biological features in this latent, so to lower this loss. And the second term is a simple classification loss to train the posterior batch distribution. And the last terms will show that it will be bounded by some constant. So this two reweighting factor is quite important in our lower bound. And we need to find some way to approximate these variational distributions. A very simple way to think about it is to concatenate the both both phenotypic latent and drug structure latent, and train a classifier based on the concatenation of the two latent. However, this have several challenges. First is lack of empirical observations. And also because in the observations that we can use, we only have access to the samples that have I equals to 1, which means that they are paired, they are positive paired. And in that case, we know what is XB1, what is the real XB1. So if we apply this also to negative samples, there will be some out of distribution generalization issues. And furthermore, if we train the classifier based on the concatenation, it will involve very computational intensive steps because we have to estimate this probability individually for each anchor positive pair or anchor negative pairs, and we can't reuse the calculated values. So due to these challenges, we choose to approximate these posterior batch distributions conditioned on both latent with a weighted average of the posterior batch distribution conditioned on single latent, as shown here with the weight, with the weight hyperparameter alpha. And we can further show that with this estimation, the second last term C will be lower bounded by 0, and and the last term is a constant. So our final loss is the reweighted clip loss together with the classification loss to train the classifier. And this hyperparameter alpha represents a trade-off between like getting access to more negatives to improve the model's generalization capability and to remove the bias or mitigate the batch confounding issue from the data. And we can also see that when alpha equals to 1, this loss will collapse to the normal influence E loss in the clip model. And compare with the previous baselines that I mentioned, our model has two kind of advantages. The first is, so compared to the clip model, it is able to remove the bias from the batch confounder. And then compared to the conditional contrastive learning method, our model is able to get access to a large supply of negatives and have better generalization capability. And also, because this posterior batch distribution, these reweighting factors are calculated within the training process, it is a very flexible adaptive adjustment with respect to both different experimental batches and also across the training process. For example, the confounding issue of each experimental batch might be different. Some experimental batches, the drugs may be very randomly shuffled and assigned to them and don't have much batch confounding issue or structure similarity within the same batch. And for some batch, this issue might be more severe. And with this posterior distribution, we can adjust for it. And also across the training process, if during the later state of the training process, the batch confounder has already been mitigated from the latent representations, then we will see that the the like reweighting factors will become constant, and the model is no longer further over reweight the different samples. And this is the figure to illustrate the pipeline of our method. The left part is the same as the clip model, where we have two data modalities and we have the encoder for each single modality. And we also have the latent representations of each of this modality. And we can calculate the similarity between each pair in this matrix, with the diagonal being the positive pairs and all the other elements being the negative pairs. However, in the right part, we can also calculate the weight matrix for each of this element in the similarity matrix. And this each weight is calculated based on the output of these two batch classifiers together with the observed batch identifier of the anchor, as shown below, which is another formula of the previous reweighting factor and the weighted average approximation in our propositions. And we do some experiments to validate our method. Oh, question.
Yes. Do you mind going back to the previous slide? Yes. How accurate are the classifier here, given that in some data set we can have very small batches and some other batches that are very large? Like, how do you deal with that?
You mean small batches and the batch number, like the number of different batches is large?
That was my second question. So yeah, in some cases also the number of batches is quite large. But the first question was, if some batches are very small, what will happen is the classifier might be just like, and some batches are larger than other, the classifier might maybe put in all the sample in one batch and instead of the smaller ones.
I see. So first in our data, because they are doing the experiment in the 384 well plate, so the distribution across different batches are quite average. and therefore we don't really adjust for this issue in our experiment. However, for some cases where there's really this bias issue, I guess it is good to reweight the classification loss based on the batch distributions, or even reweight reweight the input samples a little bit according to the biased batch distributions would help. And also during the training, I tracked the cross entropy loss of the batch classifier, and I observed that, for example, the loss is firstly decreasing because we are training the batch classifiers, and the prediction becomes more accurate. And during the training process, later in the stage, the cross entropy loss is going up slowly with the training, which means that the model is learning to remove the batch-related features during the training process.
Okay, perfect. And how do you deal with a huge number of batches that might impede the accuracy of those classifiers?
So in the two data sets that we're using, one have 100 batches and the other have 1,000 batches. And empirically, we don't really have issues for this large batch numbers. I guess maybe it's because so first the number of samples in our batches is quite average, and also the batch effect is very significant, especially in the phenotypic screening data. So even with this hundreds or thousands of different batches, like a simple random forest classifier can have a very good prediction. So I guess that is not the issue. And if there's difficulty in predicting this batch number, it means that the batch effect is not very severe, which is also good.
Perfect. Thank you.
No problem. Yeah, so now I'm going to introduce the experiments that we do. We do three kind of experiments. The first is the simulation setting to illustrate intuitively in the 2D case how what our model is doing compared to the other baselines. And this is based on some simulation data. The good thing is that for the simulation data, we can know the ground truth real effect, which is the thing that we want the model to capture. And also there are some batch effect. So we have 5 real effect and 25 different batch effect labels in the simulation setting. And we project the latent representations directly to the 2D space, so that we don't need to use other like t-SNE or UMAP to do the further projection. And this is reflecting the real distribution, the raw distribution of the latent. So compare with the CLIP and the conditional contrastive learning method, we can see that our method can separate the data by real effect very well. However, it is very well mixed according to the batch effect labels. And here for CLIP model, it can also distinguish some groups of real effect, but not all. And also we can see that it's confounded by the batch effect. For example, this green samples are all from the same batch. And for the CCL, the batch effect labels are also well mixed. However, because the negative samples are limited, we can see that the real effect labels are not well separated except for the orange class. And our model consider take both into account and have a good performance overall. And then we do some experiments in the real drug screening data. And we use 2 types of drug screening data. One is the gene expression, where we use the Linux dataset. And the other is the cell imaging, and we use the cell painting dataset from grade 2017. And to evaluate the outcome of the method, we use two types of tasks. The first is the molecule-phenotype retrieval for drug repurposing, which means that we want to identify the molecules from a retrieval library that are most likely to induce a given phenotypic change. And this is useful for drug repurposing because, for example, we know the gene expression of the healthy state and the disease state of a cell, and we want the drug can shift the gene expression from the disease state to the healthy state. Then we can encode that gene expression in the latent space, and from the drug libraries that we have in hand, we can find the several most similar drug structure in the library with respect to the gene expression latent. And then based on the similarity calculation, we can select the drugs that are most hopefully be able to utilize as the repurpose, like for drug repurposing. So here we have two kind of retrieval libraries. One I name it as whole, and the other I name it as batch. And this whole is just all the drugs in the validation set, which contain about 2,000 or 3,000 drugs. And this batch are all the batches, are all the drugs within the same experimental batch as the retrieving gene expression or imaging. So if a model only learn batch effect, it may still have good performance in whole, in the library whole, because it can propose the retrieved one based on a random sample of the drugs from the same batch. However, its performance in batch would be very bad because it can't utilize the batch features as a shortcut. And so here is the performance of our model compared to the baselines. And we can see that our model is the only one that can achieve good performance in both of these retrieval libraries. For example, our model can have similar or better performance than CLIP in the whole library and have similar or better performance compared to CCL in the batch library. And this exactly show that our model balance the trade-off between the large supply of negatives and also the debiasing. And okay, I see another hand.
Yeah, there's a question in the chat about the accuracy metric. Is it related to the cosine similarity?
Yeah, so I calculate the latent factor of both the phenotypic screening and the drug structure. And because in contrastive learning, they are already normalized. And then I calculate the cosine similarity. And this retrieving means that this top one accuracy means that the most similar drugs is the correct drug corresponding to the gene expression. And similar for the top 5 and top 10.
Yes.
Okay. And the second experiment of the drug representations is the transfer learning to fine-tune the model for some downstream property prediction task. And we select the biological related features, the biological related tasks, because we think the gene expression, like the phenotypic screening data, will help with the biological understanding of the drug structure. And here is the result. First is multi-vector corresponding to a random initialization without the pre-training. And we can see that all this pre-training method with each kind of drug screening data set have very significant improvement with in comparison to the random initialization, which indicate that this drug screening data really helps a lot of our understanding of the biological effect of the drug. And furthermore, if we compare our model with the two baselines, we can also see that it can consistently outperform the other models in most of the data set. And finally, to show that our model can be applied in a more general cases, we also do some experiment in the non-drug related domain, which is the representation fairness. And we use several standard fairness data sets, where here the sensitive attribute is no longer the batch, but it is the race and gender of this data point. And the first is the prediction accuracy for the binary prediction task, and the other three are different fairness metrics that are used in previous papers. And we can see that when achieving a similar level of accuracy, our model have the best fairness metrics as shown here, which means that it can remove the bias from race and gender the best and increase the representation fairness. And here are some of our references, and thank you. Welcome to other questions.
Great. Thank you for the talk. Are there any questions from the audience?
I can start.
Yeah.
Do you mind going back to the research session?
Yeah.
Yes.
Which results?
Yeah. So I'm not sure I understand this experiment and the next one, at least in showing the effectiveness of your method. One thing that I was expecting is, so if the batch effect has been removed accurately, like I was expecting some experiment with new batches. I trained on some batches and holding out some other ones and seeing if the generalization, at least the model that has been built on some batches generalize well to the other batches. And maybe that's what we are doing here, but I'm failing to understand it from what you explained. So do you mind maybe contextualizing this a bit more?
Yeah, sure. So these experiments are all like we don't split the train and validation set based on batches. So it's a random splitting. And I think to also test it with the splitting by batch would be a good experiment and a good suggestion. But I'm not totally sure that when generalizing to new, like when you have a new batch, it might still be possible that the model is capturing batch effect, but this batch confounding issue can also be generalized to this held out batches. Then in that case, the evaluation can also be kind of problematic. But yeah, it's good to see how it will perform if we held out some batches, especially some different batches that maybe we can pre-calculate it based on some metrics.
Yeah, because here, like, so if you head out some batches and just running the classical algorithm, I guess we can train the whole data. You can train on both the training and the test set, and then hold out some batches and learn a model to differentiate, like maybe in the differentiate between the different biological factors that differentiate the sample, and try to see if you generalize better to the other batch. Because if you have batch effect, there's a huge likelihood, and it often happens that you won't be able to generalize a model that differentiate a biological background to new batches, right? So maybe that's the test that I was proposing.
I see. Yeah, it's a good point.
Cool. And on the next figure, the next one, I'm not sure I understand why. So basically, you use this the representation that was learned on the molecule side to test on other tasks that might not be related to the data collection process to see if the representation is better.
Yeah.
And this kind of I'm not sure why you would expect some improvement here compared, because the number of molecules that you have is somehow limited compared to some other self-supervised or unsupervised training method that we train with a million of sample and hope for from generalization of the representation.
Yeah, so first here I don't really compare with the with those pre-training methods with self-supervised training, because first the training sample, as you mentioned, are very different. We use about 20K or 30K drugs for pre-training, and they use like millions of drug structures for pre-training. And also we utilize the drug screening data, and they didn't utilize the drug screening data. So there's actually no fair comparison. And speaking of that, for the drug structure encoder, I'm also use the multi-vec Fisherizer or the multi-vec pre-trained embeddings instead of training a gene, for example, from scratch, because it can better generalize to new drug structures. So I guess our model and this multi-vec Fisherizers can also be replaced by other pre-trained self-supervised or unsupervised molecular encoders very easily. And I would also expect a similar or good performance. And this molecular property prediction is a very out of distribution task, because all these molecules are not most of these molecules didn't appear in the drug screening data set that we are using. And there may be very huge distribution shift. However, because these are all biological factors, like some toxicity or side effect, so we would expect that our model trained with the phenotypic screening data will have more information of the biological effect of the drug, and therefore have some improvement on this data set. And also, as shown here, we do have some information gain with the additional phenotypic information screening data. And to compare with the previous models, I think it's mainly we mainly want to see how much real biological features that this model is able to capture. And this properties is one of the ways to measure this. And I guess there are some other maybe more relevant properties from other paper and data set, and I'm going to test it. I haven't done it for this paper, and just use this quite benchmark data sets for testing in this paper.
Cool, thank you.
I have a question about p-hat. I'm wondering if there wouldn't be an alternate representation choice that could make sense, which is just, you take those embeddings, maybe you project them to a new space, but you take their like cosine similarity, or like, clipped inner product as the probability, rather than this weighing, which suggests that they're like independent but have a particular weight. I feel like you'd also be able to deal with OOD because you're still forcing these two vectors to come together in different sources. Is that something you thought about or tried?
I see. Can you see more specifically how do you calculate this p-hat based on the latent in the latent, like in the cosine similarity in the latent space?
So you had like these two classifiers in the, I think, figures, if you slide. these could just be like, let's say MLPs, and then to calculate MIJ, you take like, I don't know, the sigmoid of the cosine similarity of those two vectors, right? And then you still get a prediction where the two bits are not independently added together. There's still some correlations that can occur.
I see. Which is like calculate the geometric average instead of this normal average of these two classifier output.
No, I mean it's okay if you haven't tried, but I guess I'm wondering actually, like, it feels like a pretty strong assumption of independence to have these two things calculated separately. Is that something you've investigated a bit?
Yeah, I agree. And there are, we do try a little bit of different method, like different ways to approximate this variational distribution. And that cosine similarity way, I think it also, so the cosine similarity is the product of these two posterior batch distributions. And I think we finally choose to use this average because in the product case, there's still the assumption that if, for example, one of the p-hat outcome is 0, even if the other one is large, it will also be projected to 0. And that is something that is not quite desirable. However, I'm not totally sure whether I tried that or not in my previous experiment. But if I remember correctly, they give not very different outcome.
Right. Do you think that's an issue with like the data, or is just like learning this a bit hard?
I'm not sure. Yeah, there is more to think about in this approximation part.